started with reviewing for the optional final exam. So we started off learning about classification and the 3 domains of life. Remember, 2 of the 3 domains are prokaryotic. We have the bacteria and the archaea. And the last domain is eukarya, which has all the eukaryotes in it. And the bacteria, of course, belong in the domain bacteria. And we learned that domain was the largest grouping, followed by kingdom, phylum, class, order, family, genus, species. So there were 2 competing hypotheses concerning the origins of these microorganisms. One of them was spontaneous generation, life arising from non-living matter, and the other was biogenesis, life from life. And we talked about several scientists and their experiments like John Needham, Spallanzani, Louis Pasteur. You know, they boiled broth. Needham did it wrong. He either didn't boil long enough, or he then transferred his boiled broth to a non-sterile container, so he contaminated it. Microorganisms grew. Spallanzani did everything correctly, but no one believed him. And 100 years later, Louis Pasteur repeated the same experiments over and over again using his special S-shaped or swan-necked flask, which was open to the external environment, which meant that air and the mysterious vital force could get in, but nothing grew until Pasteur tilted his flask and contaminated the broth. So finally, Louis Pasteur conclusively demonstrated microorganisms come from pre-existing microorganisms, or biogenesis. 3 most common shapes are rods, cocci, and spirals. The arrangements, if you have cells stuck together in a chain, we call that strepto. So streptococcus, streptobacillus. If they're clustered together, that's staphylo. If we have pairs, that's diplo. And if you have clusters of 4, what did we call clusters of 4? What do we call a cluster of 4? A tetrad, right? A tetrad. We know there are 2 major groups of bacteria, gram-positive and gram-negative. Remember, gram-positive bacteria have that very thick layer of peptidoglycan, and the gram-negatives have thin peptidoglycan, but they have a second membrane on top of that. So the gram-negatives have 2 membranes. And it's the gram-negatives that have the endotoxin, that lipopolysaccharide or lipid A portion of their membrane, which is toxic, but remember, it's not very toxic. The LD50 is high, which means you need a lot of it to really be toxic, so it's not very toxic. And gram-positive rods, what do they produce that's special? What do gram-positive rods produce that's special? Nobody remembers what's special about gram-positive rods? What you should always think of when you see a gram-positive rod? Spores, right? Spores. Remember, spores are survival structures, and the 2 major groups of spore-forming bacteria are Bacillus and Clostridium. Bacteria can move. We know they can swim using flagella only. So if they have flagella, and they have only 1 flagellum, we call that monotrichous. A flagellum on each end is amphitrichous. A cluster of flagella is lophotrichous. And if they're covered with flagella, that's peritrichous. When it comes to energy production, we're talking about ATP formation. And we know that bacteria can be or can use aerobic respiration or anaerobic respiration. For aerobic respiration, the process starts off with glycolysis. Remember, 2 ATPs are required to start the process of glycolysis. By the end of glycolysis, the cell has produced a total of 4 molecules of ATP, but because they required 2 of them to start glycolysis, we always refer to the net production of 2 molecules of ATP. They also produce 2 molecules of NADH and pyruvic acid. The NADHs are carrying electrons to the electron transport chain to make ATP. So the pyruvic acids cannot directly enter into the Krebs cycle. They must be decarboxylated and converted into acetyl coenzyme A. At that step, no more ATP is made, but NADH is made, and 2 NADHs get made, and also carbon dioxide comes off. Now the acetyl coenzyme A is ready to enter into the Krebs cycle. Remember that glycolysis occurs in the cytoplasm of all cells, and the Krebs cycle occurs in the mitochondrion of eukaryotes, but cytoplasm of prokaryotes. And so the acetyl coenzyme A is going to cycle through many different intermediate molecules. In the process, 2 molecules of ATP are made, 6 molecules of NADH, and 2 molecules of FADH2. All the carbons get released as carbon dioxide. So the cell's only made a total of 4 molecules of ATP, but the cell has made a lot of NADH, 10 molecules of NADH and 2 molecules of FADH2, and they both carry electrons to the electron transport chain to make ATP. So the electron transport chain must be embedded in a membrane. For prokaryotes, it's the cell membrane, the plasma membrane. For eukaryotes, it's the inner mitochondrial membrane. But when the electrons are donated to the complexes in the membrane, as the electrons are passed from complex to complex, hydrogens get pumped across the membrane. Those hydrogens accumulate in a high concentration. We know that molecules in a high concentration want to diffuse to an area of low concentration, which in this case is on the other side of the membrane. But the hydrogens cannot get back through the membrane by themselves. They need help. That help is a special transport protein, ATP synthase. So that's the enzyme that makes ATP. As the hydrogens flow through, move through the ATP synthase, that's the alternate form of energy that ATP synthase uses to make ATP. Remember, we call that the proton motive force. That's also the chemiosmosis part of cell respiration. And so that's where cells make the majority of the ATP. And remember, the process is aerobic cell respiration because oxygen is the final electron acceptor. At the end of the transport chain, the electrons have to go somewhere, and they combine with hydrogen and oxygen to make water. So water is a metabolic byproduct. But that process is aerobic. Of course, we know many microorganisms can perform anaerobic respiration, and the one type we talked about was fermentation. So fermentation just uses glycolysis, no oxygen. Different kinds of end products can be produced, right, by fermentation. But the cells are limited to 2 molecules of ATP per glucose because fermentation just uses glycolysis. If you're a single-celled microorganism like bacteria or yeast, that's fine. You can live on 2 molecules of ATP per glucose. But if you're a complex multicellular organism, you can't live on that. You can't do work with so little ATP. So we really do need the oxygen, right, for complex multicellular organisms. Now, oxidation and reduction, remember when an atom or molecule loses electrons, it becomes oxidized. When an atom or molecule gains electrons, it becomes reduced. Whenever there's an atom or molecule that loses electrons, there's some other atom or molecule to gain them. So oxidation and reduction reactions are coupled together, and we call those redox reactions. So this doesn't convert well, but DNA to RNA to protein. We know that DNA is transcribed into RNA, which is translated into protein. And so we know the enzyme that makes RNA from DNA is RNA polymerase. Ribosomes read the messenger RNA to translate it to make proteins. And when DNA is copied, we call that replication, and that's accomplished using DNA polymerase. Both DNA and RNA polymerase enzymes can only add nucleotides in one direction, the 5' to 3' direction. So the flow of information in a cell is the central dogma of biology. And is it always just a one-way flow? Is there any way we can go backwards? Where and how? Anybody remember where and how we can go backwards? Everybody remembers that we can make DNA from RNA. We can reverse transcribe RNA back into DNA using the enzyme reverse transcriptase, which certain viruses like HIV have, right? So when it comes to genes, eukaryotic genes are more complicated. Eukaryotic genes have introns, those non-coding regions. And so exons are the coding regions, the ones that will be ultimately translated to make proteins, and the introns are typically spliced out. So once the initial messenger RNA transcript is made, the introns get spliced out, and then you have the final messenger RNA transcript. Prokaryotes don't have introns, at least not the same kind that eukaryotes have. So that presents a problem if you want to clone a eukaryotic gene into a prokaryotic cell, right? Because if the introns are included, the bacteria cannot remove them. And so the protein's not going to be functional because it has a lot of extra amino acids in it. So if you want to clone a eukaryotic gene into a prokaryotic cell, remember you must isolate the processed messenger RNA transcript with all the introns removed, use reverse transcriptase to make a DNA copy. You can insert that gene into a plasmid and then use the process of transformation to get that plasmid inside the bacterial cells. When it comes to regulating gene expression, bacteria use operons. Remember, the operon is the unit of regulation for prokaryotes. So if you have multiple enzymes in a metabolic pathway, there are multiple genes. All those genes are clustered together under the control of the same promoter. So when that promoter gets turned on, all those genes are transcribed and translated to make all the enzymes needed for that metabolic pathway. In between the promoter and the structural genes is the operator sequence. That's where the regulation occurs. Remember, a repressor protein binds to the operator, and that's what helps to keep that operon turned off or to turn it off. So we said there are two major types of operons, inducible and repressible. Inducible operons are normally turned off because they code for enzymes to use different substrates for energy. So inducible operons code for catabolic pathways. So if a particular food source is not needed or not available, that operon is turned off. It gets turned on when the cells have run out of glucose. There's no more glucose, and so those cells have to look for something else to eat. If there's another food source like lactose, then the lactose operon gets induced, it gets turned on. But only if all the glucose is gone and if lactose is present. Once all the lactose is used up, then the cell will turn off the lac operon and look for something else to eat. When it comes to biosynthetic pathways, anabolic pathways, those are encoded by repressible operons. These are normally turned on, and they have to get turned off only when needed. So when the cell has made enough of a particular product, then the operon gets turned off. This saves a lot more energy than feedback inhibition. Because remember, with feedback inhibition, one of the enzymes in the metabolic pathway gets turned off. In the case of a repressible operon, the genes get turned off, so they're not transcribed and translated, so no enzymes get made, and that saves a lot more energy. When it comes to horizontal gene transfer, we know that bacteria can do this using transformation, conjugation, and transduction. So we talked about a transformation. It happens naturally with short linear pieces of DNA where bacteria can absorb those linear pieces. In the laboratory, we use plasmids, so we have to force the bacteria by making them competent with heat shock and calcium chloride. But transformation is one way that bacteria can acquire genes. Conjugation is another. Remember, there are two cell types that conjugate, F plus and HFR. They contain the fertility gene, which makes a pilus. They conjugate with an F minus cell. When an F plus conjugates with an F minus, a copy of the fertility plasmid is transferred. So the F minus cell becomes F plus. When an HFR conjugates with an F minus cell, the F minus cell stays F minus because the HFR has an integrated plasmid. And so it still makes a pilus, but when a copy of DNA is sent over to the recipient cell, it's a copy of the bacterial chromosomal DNA. And normally, the bacteria do not stay hooked together long enough to allow much DNA to be transferred. So the fertility plasmid, the integrated fertility plasmid, is rarely, if ever, copied over. So the F minus cell stays F minus, but it has acquired some bacterial DNA from the donor cell. So it may have acquired a new characteristic. And when it comes to transduction, that's mediated by a virus, where the bacterial viruses, the bacteriophages, can carry DNA from one host cell to another. But all three of these mechanisms of horizontal gene transfer allow for bacteria to acquire new characteristics within the same generation. So the bacteria do not need to mutate and evolve a new trait, they can simply acquire it. We identify bacteria initially by staining and microscopy. Then we can start performing biochemical tests, which are looking for the presence of enzymes. We know that enzymes are there because we find the end products of the enzymatic reaction. We can get more sophisticated and perform serological tests, which involve antibodies and antigens, agglutination tests, where we see clumping, Western blots, where we separate proteins in a gel and then use the antibodies to identify those proteins. We can get more sophisticated and do DNA fingerprints, where we cut up our DNA with restriction enzymes, run it on an agarose gel, and we get a banding pattern. And so we can use those patterns, right, the fragment patterns, the banding patterns, the fingerprints, to identify different organisms. If you don't have enough DNA, you can use the process of PCR to make more DNA, right? All you need is your template DNA to be amplified, a thermostable DNA polymerase that came from a thermophile, all the nucleotides, ATGC, and the primers. Remember, the primers allow for the specificity of the amplification because the primers only bind to the region to be amplified, to the gene of interest. And that's the only place where there's double-strandedness. And so the DNA polymerase is only going to hop on to that double-stranded region. We talked about Koch's postulates. So Robert Koch, he came up with a series of steps used to identify the cause of a particular disease. And we discussed how his steps have problems. Step number one, or postulate number one, the suspect microorganism is only found in diseased individuals, never healthy ones. We know there are exceptions, right? Carriers are healthy, yet they're carrying a pathogen that they can transmit to others and make other people sick. Sometimes our normal flora can become opportunistic pathogens, and we all share mostly the same kinds of normal flora. But assuming that the suspect microbe is only found in diseased individuals and not healthy ones, we should be able to culture that suspect microorganism in the lab. Again, this is not always possible. We don't know the growth requirements for every single pathogen. But assuming that you can culture that suspect microorganism, you then use that culture to infect a test animal. And that test animal should develop the same symptoms. Again, this is not always possible. Many pathogens are very host-specific, so we may not be able to find an appropriate test animal. But assuming we do, and your poor little test animal gets sick, you then should be able to re-isolate that suspect microbe actively reproducing inside that test animal. And if you can do that, you can conclusively demonstrate the cause of a particular disease. And we call the study of diseases, the cause of diseases, etiology. We talked about fungi, they're eukaryotes. That's one of the reasons why it's very difficult to treat fungal infections, because they are so similar, right, to our cells at the molecular level. But remember, we discovered ergosterol, a membrane component important in fungal cell membranes that we do not have. But we said that we classify fungi based on the sexual phase. And we talked about several major phyla: Ascomycota, Zygomycota, Basidiomycota, Deuteromycota, Oomycota. Remember, if we could not find the sexual phase, then the fungus was put into Deuteromycota until such time as usually the sexual phase is discovered. Oomycota is no longer in the kingdom fungi. It's been reclassified as Protista. A fungal infection is a mycosis, right? When it comes to bacterial infections, we use antibiotics, which technically are chemical substances produced by one microorganism that inhibits another microorganism. So antibiotics are naturally occurring. We do have synthetic and semi-synthetic versions, right, that humans have created. But the problem with antibiotics is resistance. So there are many potential targets in the bacteria: the cell wall, which is the most selective target, the cell membrane, DNA replication, transcription, translation. Remember, there are many different kinds of antibiotics that target the ribosome, like tetracycline, streptomycin, chloramphenicol. You could find a metabolic pathway and block it. So there are many potential targets. But sometimes the bacteria have mutations in that target, so they become resistant. And so bacteria can mutate to become resistant. We know that there's always some resistance present in a population of bacteria as the result of random chance spontaneous mutations. Sometimes bacteria can acquire resistance from other bacteria. Maybe they acquire the gene for efflux pumps, so they're able to pump out the antibiotics so fast the antibiotic's no longer toxic to them. So there are many different possible mechanisms of resistance. When we use antibiotics, we select for those resistant mutations. And if we use antibiotics inappropriately, right, not by prescription, if we don't take all of our antibiotic when we're sick, So we are helping to create, right, these resistant bacteria. It's because we're abusing and misusing antibiotics. Some other chemicals that we use to kill microorganisms are antiseptics and disinfectants. We know that antiseptics are chemicals used on living tissue. Disinfectants are used on inanimate objects and surfaces. We said sometimes they can be used interchangeably. Not always, but sometimes. Things like isopropyl alcohol or hydrogen peroxide, they can be used as either an antiseptic or disinfectant. But something like bleach, you never want to put bleach on a cut or a scrape, right? So sometimes they can be used interchangeably, but not always. More contact time, longer exposure time makes that chemical agent more effective. You also have to take into account microbial characteristics. Are you trying to kill spore-forming bacteria? Are you trying to kill bacteria with unusual cell walls like Mycobacterium, which is thick and waxy? Viruses are not living organisms, right? They're not cellular organisms, no cytoplasm, no organelles, no ATP production. They're basically a piece of nucleic acid, either DNA or RNA, surrounded by a protein coat called a capsid, with or without a lipid membrane. If they have a lipid membrane, it's because they stole it from the host cell by budding or exocytosis. When it comes to bacteriophages, right, bacterial viruses, remember the capsid and the associated proteins stay on the outside of the cell. Only the DNA gets injected into the bacterial cell. And at that point, we'll follow the 5 typical steps of viral replication. So attachment is the first step for any virus. With bacteriophages, they attach to cell surface receptors and use lysozyme to drill a hole to the cell wall and inject the DNA. Remember that animal viruses are engulfed completely into the cell, usually through receptor-mediated endocytosis. Once inside, the bacteriophage DNA will take over the host cell. It'll force the bacterial cell to replicate the viral DNA, transcribe and translate it to make viral proteins. That's the biosynthesis, and the maturation and assembly to form new virus particles, where the DNA gets incorporated into the capsid, and then bacteriophages use lysozyme to rupture the host cell wall. Animal viruses can also rupture the host cell wall, or they can bud and steal host membrane. Bacterial viruses that kill the host cells are lytic viruses. Lysogenic viruses are dormant bacteriophages. Remember that in the case of lysogeny, the viral DNA will circularize and integrate with the bacterial chromosome, forming a prophage. At that point, it stays dormant. It can stay dormant for generation after generation as the bacterial cells multiply. But at some point, that prophage can leave. It can cut itself out. That could be caused by a random chance event, UV, certain chemicals, or temperatures. And if the prophage cuts itself out, it follows a lytic cycle and will kill that host cell. Remember, we said there are some consequences of lysogeny. One of those consequences is that the prophage may carry a toxin gene, which makes those bacterial cells pathogens. So those bacteria are only pathogenic because they're infected with a phage, which carries a toxin gene with it to make the bacterial host sick, so the animal sick. Latent viral infections, that's the term we use for animal viruses which go dormant. Best example is herpes. If you're infected with herpes, remember you're infected forever. It can go dormant, you're asymptomatic, and then the virus can reemerge. And then you have symptoms. And then it can go back and hide again. And remember, it hides in nerve cells. HPV stands for human papillomavirus. Remember, that causes warts. It has that special protein to attach to one of your chromosomes so that every time your cells divide, every daughter cell is infected with HPV DNA. Herpes, as I was just mentioning, is a latent infection which periodically reemerges, where you have symptoms, and then you become asymptomatic again. And that's because the herpes viruses are hiding inside nerve cells where the immune system cannot get to them. HIV, as we mentioned, is a retrovirus. It uses reverse transcriptase. When it forms a provirus, where the integrated HIV DNA is part of one of your chromosomes, it's a permanent part that never leaves. So it's one of the problems with getting rid of HIV is that you have so many cells infected with the provirus, you cannot eliminate those cells because the overwhelming majority of those cells are immune cells like helper T cells, which you need, right, for the proper functioning of your immune system. We mentioned that TB has a relationship with HIV. Remember that approximately 90% of people infected with TB have dormant TB, so it's not active, and those people are healthy. But if they become immunocompromised in some way, then the dormant TB can reactivate. And so HIV infection is what caused the reactivation of dormant TB to become active TB. So as HIV spread, more and more people got infected with HIV, the rates of active tuberculosis increased significantly, at least through the 80s and 90s and into the 2000s. When it comes to disease transmission, we have direct contact, right? Touching, kissing, sexual intercourse. Indirect contact using fomites, inanimate objects like doorknobs, like keyboards, cell phones, things like that. If you cough or sneeze, you produce droplets. So a droplet infection generally only spreads about 1 meter or so. It depends on, the wind conditions, environmental conditions, things like that. If you want to transmit disease over a greater distance, you need food and water, or you could have biological vectors and insect vectors to transmit diseases over greater distances. Incidence and prevalence. Remember, incidence refers to the number of new cases of a disease relative to population size and indicates the risk, right, the relative risk of getting infected or contracting that disease at that particular point in time. Prevalence is both old and new cases relative to the population and can indicate the burden of that disease on that population. Pathogenicity refers to the development of disease, right, because pathogens are disease-causing organisms. Virulence is a function of infectivity and toxicity, right? The ability of your pathogen to overcome host resistance, to overcome the immune system. And toxicity, how much damage does it cause to those cells and to that host? Many pathogens regulate virulence with quorum sensing. So remember, once the pathogen gets inside of you, it starts to multiply, and the bacteria secrete those chemical signals. And so when they sense they have an army of cells, they all launch virulence simultaneously. In that way, they can overcome a large host. Your immune system responds to antigens, anything it considers foreign, right, to the body. Haptens are antigen wannabes, right? They're small and insignificant, but when they get stuck to one of your tissue proteins, that combination becomes an antigen. If a pathogen is trying to infect you, we have 3 lines of defense. The first 2 lines are non-specific. So your first line defenses are things like skin, stomach acids, mucus and cilia, things like that. And they're non-specific, right? So they work against a wide variety of pathogens. Your second line defenses can involve phagocytes, right, which wander around and eat things. Inflammation is a second line defense, which helps bring your immune system to that site of injury or infection. Fever is a defense, which also helps to inhibit pathogens and stimulate the immune response. And if a pathogen gets through the first and second lines of defense, you have that last line, your acquired immune response, which is very specific, right, to that particular pathogen. So your immune system is going to make a response that specifically targets that pathogen. And that involves T cells, B cells, and macrophages. Remember, macrophages wander around, find things to eat, digest them, present the antigen to a helper T cell, along with the major histocompatibility complex. The macrophage also secretes interleukin-1 to stimulate helper T cells. Helper T cells secrete interleukin-2, which stimulate themselves, as well as stimulate B cells and cytotoxic T cells. Remember, B cells, when they're stimulated, they will proliferate and differentiate into memory cells and plasma cells. The plasma cells secrete the antibodies, and the memory cells remember for later. Cytotoxic T cells can destroy cancer cells or cells infected with viruses. If your T cell count goes way down, you become immunocompromised, right? Your immune system is not able to function very well. Now, your B cells make a lot of antibodies. And we talked about the 5 different types of antibodies: IgGs, IgMs, IgA, IgD, and IgE. The IgG is the most common type of antibodies you find in the blood, but they are typically made upon those secondary exposures. So the memory cells you have that remember for later, they will generate lots of IgG antibodies. But the first time you encounter that antigen and make antibodies, you make IgMs, right? Those big pentamers with all those antigen binding sites. IgA antibodies are dimers found on mucous membranes. IgDs are found in lymph and are attached to B cells and act like a receptor, right, to bind to antigen and stimulate that B cell to make antibodies. IgE antibodies are involved in hypersensitivity reactions, but their main function is to help to destroy larger parasites. And antibodies are so very important because they do many things. They can neutralize a virus or a toxin. Remember, viruses must attach to host cells, so they have attachment proteins or spikes they use to attach. If the spike is covered with antibodies, that virus has been neutralized because it cannot attach. A toxin, if an antibody binds to a toxin, that toxin is no longer toxic, so it has also been neutralized. Agglutination is where antibodies are used to clump pathogens together, so they can help to clump together a bunch of bacterial cells, and it's easier for a phagocyte to eat that clump rather than to eat each cell individually. A precipitation involves soluble antigens. The antibodies bind to the soluble antigen, it becomes insoluble, it precipitates, it's solid, now a phagocyte can eat that. Opsonization, when something is covered with antibodies, that enhances and stimulates phagocytes to come eat it. Complement activation, remember the complement cascade results in proteins that assemble and poke holes through cell membranes. I also mentioned the antibody-dependent cell-mediated cytotoxicity, the ADCC, which is used against larger parasites like worms. Again, all those antibodies attached to it help to attract a variety of cells to attack and destroy that large parasite. So antibodies are crucial to the normal functioning of the immune system. If you do have memory cells, then you have immunity. So remember that it takes 7 to 10 days, right, to make antibodies the first time you're exposed, but you have those memory cells, the memory B cells, memory T cells. So that means that you can respond as quickly as a day the next time you encounter, right, that particular pathogen. So that gives you long-term protection, usually for your entire life. But we mentioned that many pathogens do mutate on a regular basis, so they change just enough that the antibodies you already have are no good, and so you get sick all over again. But you have active immunity if you actually are making the antibodies. It's natural active immunity if you make those antibodies in response to an infection. It's artificial active immunity if you make those antibodies in response to a vaccine. So it's still active because you're making them, but it's artificial because it's a vaccine. Passive immunity occurs when you receive foreign antibodies. So they're not yours, you're not making them, very short-term protection. So it can be natural passive immunity, as in the case of mom's antibodies crossing the placenta, or in the colostrum, right, the first milk produced by mom. It's considered artificial passive immunity if you're receiving antibodies made in another animal. So we talked about how if you got bitten by a poisonous snake, you could get an injection of an antivenom, and those antibodies would neutralize all the snake venom, so you survived. But these are short-term, right, treatments, so the antibodies are not going to last, and you're not able to produce more of them because you did not make them to begin with, so it's passive. Privileged sites and tissues. Privileged site, the immune system does not go there. Because if the immune system went there, it might see something that's foreign, like sperm, and destroy them. Or if you have an immune reaction like inflammation, that can cause permanent damage, right, to places like the brain and the cornea. So the immune system does not go there. A privileged tissue, the immune system goes there, it sees the tissue but ignores it. So things like a fetus, half the antigens of that fetus are foreign, but the immune system, and mom's immune system ignores that fetus. Heart valves, the Achilles tendon, right? The immune system sees those things but doesn't care. Hypersensitivity reactions occur when the immune system overreacts, and the overreaction can lead to damage or death. We talked about 4 or 5 types of hypersensitivity reactions. Remember, type 1, 2, and 3 are mediated by antibodies, and type 4 is T-cell mediated, so it's slow, it's delayed. Type 1 are the anaphylactic type of reactions, so they happen quickly. They can be localized or systemic, so they can potentially be life-threatening. Type 2 is cytotoxic. Type 3 is the immune complex type. Type 4, as I said, is T-cell mediated, often involving haptens. Type 5, we said was stimulatory, but we normally classify those as type 2 reactions. But hypersensitivities are overreactions by the immune system. They can also cause autoimmune diseases, right, where the immune system attacks the host or attacks a transplant in that host, right? So transplant rejection is a big problem because of the immune system. So that's basically all I have for review. Any questions you have about the lecture exam? It's hard to summarize an entire course in 50 minutes. No HIV. I talked about HIV, right? What do we need to know about HIV? Retrovirus, RNA genome, reverse transcriptase, increase the rate of TB. Anything else, Christiana? All right.